DNA is wound in a double helix structure composed of nucleotides, the basic structural unit of the genetic molecule. Each is made up of a phosphate group, pentose sugar, and a nitrogenous base. There are 4 types of nitrogenous bases. Adenine and guanine are the purines. Thymine and cytosine are the pyrimidines. Adenine bonds to thymine, and guanine bonds to cytosine, using hydrogen bonds. Phosphodiester bonds bind these nucleotides together, acting as the backbone of the double helix structure.
The human body is made up of trillions of cells, each containing 46 chromosomes that contain the genetic information necessary for cell function. The first step of protein synthesis is transcription, in which messenger RNA is produced from a segment of DNA. Transcription starts with the helicase enzyme unzipping the DNA by breaking the hydrogen bonds between nitrogenous bases. It is aided by topoisomerase, an enzyme that unwinds the double helix structure, relieving tension ahead of the replication fork. RNA polymerase synthesizes a primer in the 5' to 3' direction and allows the polymerase to transcribe the DNA into RNA. C goes to G, T goes to A, and A goes to U. The strand that is produced is messenger RNA, which is sent out of the nucleus into the cytoplasm, where it meets a ribosome to initiate translation. mRNA attaches to the ribosome, where one of its codons is brought a corresponding anticodon by tRNA. This process is continued on the rest of the mRNA, where the amino acids, or building blocks of proteins, corresponding to the anticodon attached to the tRNA are bonded by peptide bonds and form a polypeptide.
Virus basics. An invading virus will bind to the receptors of a cell to trigger endocytosis to enter the cytoplasm. The virus then releases its contents and enters the nucleus, where its RNA hijacks the reproductive system of the host cell by its processes of transcription and translation to replicate copies of the virus that will travel off to different cells. However, white blood cells detect the foreign antigens on the virus's surface and immediately activates plasma cells that will secrete large quantities of antibodies to kill the virus and develop memory cells that remain dormant until a future attack by the same virus. Vaccines work similarly in the way that they inject antigens of a virus into the blood, and the body will react by producing an immune response and keeping the proteins in the immune memory in case of a future attack where it needs to release antibodies for that same virus.